So Gray is about to share with you what we have learned the hard way by doing something over and over and over again, meaning optimizing our instrument parameters and diagnosing what went wrong. Because at the beginning when we started, most of the time things went wrong. We did not have pure spectra. We had very dirty spectra. We did not have high identification rates. We did not have the expected intensities in the various labeled samples. And I found myself writing the same scripts over and over again, doing the same plots over and over again. And at some point we decided that it's much more efficient to bundle all of these ideas, so that all of the things that we have found to be very useful for us, into a software package that we can make easy for everybody else to use. And Gray, a graduate student in my group, led the development of that software package and brought it to heights beyond anything that I was going to do on my own. So he is going to tell you about these efforts now and walk you through how you might start using it as well for optimizing your LC-MS. And not only for that, but also for sharing your data with collaborators so that you can give them a quick impression of what is the performance of your analysis at the various levels at MS1, MS2, identifications, and so on. And of course, you can use it much beyond single-cell analysis. You can use it for any LC-MS analysis. So, Gray, I'm looking forward to your talk.
So I want to welcome everyone back from their coffee break. We're going to be on the third workshop in today's workshop series. We'll be looking at optimizing your LC-MS system using a software platform that we developed in the Slavov lab entitled DoMS. And DoMS stands for Data-Driven Optimization of Mass-Spec Methods. And what I'm going to be presenting here is not so much a formula, but rather a way of approaching optimization on your own instruments and your own samples, and a tool that you can use to do this. Because no two samples are equally alike, no two systems are alike, and so it's very important that you have a toolkit that you can adapt to your own purposes. So by now you've seen this diagram a couple of times. And we got a sense of design considerations from Harrison, internal and external validations that we would want to include in our single-cell sets. Ed walked you through the process of actually preparing your single-cell samples. And what we're going to spend some time on now is this far right-hand corner, which represents a feedback loop. In terms of optimizing your mass spec, you're going to have some parameter sets that you will want to test out. You will process your data, and then you will make hypotheses and test your hypotheses as to whether or not the parameters that you varied, injection times, gradient lengths, et cetera, allowed you to maximize a parameter of interest. What that parameter of interest is depends on you. Is it the number of identifications that you have across the individual cells in your set? That sounds like a reasonable parameter to optimize. Is it retention widths? It all depends on you, and we hope that this platform that we've made allows you to achieve that aim. So we're going to approach the parameters that are inherent in each part of this data analysis pipeline. First of all, if we take a look at the actual sample preparation component, there are a lot of things that you might want to test out on your own. For instance, the carrier channel composition. For our sets, we find that twin 50-cell carrier channels seem to work reasonably well in terms of giving us good quantitation in our single-cell channels. But every different set or every biological system is different. And so you might experiment with using twin 100-cell carrier channels or lower carrier input, or as Harrison mentioned, spiking in particular peptides that you'd like to detect in your single-cell channels. Those are some parameters you'd like to vary and be able to see the effect of. Additionally, we can take a look at the parameters you can use in liquid chromatography and on the mass spec itself. So this is what your sample looks like as presented to your mass spec by your chromatography system. Chromatography is amazing. It allows us to separate out our sample in time, reduce the complexity that's being presented to the mass spec for analysis, and in our case does so based off of molecule size and also hydrophobicity. Additionally, you can see this landscape is composed of an M/Z component, the mass to charge component, and one part of this that's kind of neat to think about is that a peptide sequence can appear in multiple different slices of this M over Z landscape, depending on its charge state. If we take a peptide that has maybe a hypothetical peptide with a mass of 1,000 mass units, and it has a charge state of 1, it'll be present in the high mass to charge ratio element of this landscape. If it has a charge state of 2, it'll appear at the 500 M over Z unit slice. So it's neat to think about the ways in which chromatography and the mass spec itself allows us to kind of view our sample differently, the complexity inherent in our sample. If we zoom in a little bit, we can start to see how the individual peptides and molecules of interest kind of meet our mass spectrometer. This doesn't happen in a binarized fashion. It's not a simple like light switch on or off, but it follows this smooth increasing curve up to an apex and a decrease. And I mention this because this is something we might want to optimize. You're sampling these elution curves. You're not typically sampling the entire curve, but you're getting a slice of it that you're submitting for fragmentation. And so one thing we might want to optimize, and which we'll talk about a little bit later, is how to optimize and sample this elution curve more reproducibly at its apex. Because for low input samples, for single cell samples, you do have lower input. And you want to make sure that you're maximizing the ratio of ions that you're sampling from your analyte of interest over the amount of ions you're sampling from contaminants that are nearby in M over Z RT space. So we'll talk a little bit about that later. Additional things you might want to try out are different gradient lengths, whether or not you want to stretch things out in space or try and make your peaks sharper. Different column systems. We've explored many different columns in our laboratory, and so this a system like DoMS has allowed us to compare these and draw conclusions as to which ones perform the best on our single cell sets as opposed to maybe a bulk level input. Additionally, you can also feel free to experiment with different buffers. Perhaps you've read that a certain buffer will allow for better ionization of your peptide species. And indeed, you'll be able to use DoMS to figure out whether or not this is the case. Now we're going to take a quick walk through some parameters of interest with the mass spec, courtesy of a video from Thermo. So we see our analytes being ionized. They're traveling into the mass spec through the heated capillary. These different species are visualized as different colors, passing through a series of lenses. And they're going to travel around the curved flatopole and pass through the mass filter. Now at this point you can choose different M over Z ranges that you would actually like to submit for further analyses. The length of time that you collect these ions, this is a parameter that you could experiment with. And then these will be submitted to the Orbitrap momentarily, where we'll actually be able to get the M over Z ratios of these precursor ions. So from this, we have another very important decision that we can make that will affect the types of peptides that we'll identify downstream. Here we see 3 unique precursor species, 3 unique M over Z ratios, and we can make a decision about which ones of these we want to submit for fragmentation, which ones of these we want to try and have a reasonable chance of identifying. And this takes time, because for each dependent scan that you do, you may be missing other peptides that are being ionized and entering your mass spec. So this is a key consideration that you'll have to make. Likewise, the dependent scans are parameter rich as well. Again, we see a different group of ions entering the mass spec, traveling through our lenses. And in a moment, we'll get to see the mass filter in action, selecting that precursor species that we decided upon from the survey scan. So we have all our different ions. We have only this single precursor species remaining. It's going to enter the collision chamber. And in the collision cell, you have a determination to make about what's your fragmentation energy going to be. Are you going to bias this more for fragmenting your reporter ion? Do you want to get more diverse fragments from your precursor species? Considerations that one has to make. Collection time, again, for the fragments is another consideration. And then hopefully, you've submitted your fragments for analysis on the Orbitrap, and you'll be able to use a software package like max_quant downstream to identify this peptide from its fragments. So I show this mostly to lay out sort of the very parameter-rich space in which we work. And this is just another visualization of this as seen if you're a thermo user. This is within Excalibur. You're able to see all these different parameters you could adjust. Often you'll be adjusting multiple of these at one time. So the space is combinatorially very large. So what would you optimally need in order to make sense of all these parameters that you're adjusting? You'd need information from every level of analysis. You'd need information on how was your sample prep. You'd need information on how did your chromatography system perform. You'd need information on how you were sampling your ions. And luckily, you have access to all this information. Whether or not you're using PD or max quant, this is all provided to you. You just have to know where it is in the output data files. Additionally, you'd want to minimize analysis variability and time spent performing one-off evaluations. This is particularly important if you're working as part of a team. Sometimes people have, through no fault of their own, different ideas about which metrics are the ones to compare. And you can have a little bit of variability in your analysis just based off of the sheer number of metrics that are available to look at. So by making this reproducible, you can ensure that the variability you're seeing is due to those are the parameters you're choosing, not due to analysis. Additionally, you'd want to be able to diagnose issues as specifically as possible. And that comes back to breaking things down on the level of where they occur in the system. So we took these considerations in mind, and based on these considerations, we developed DoMS, which is a way of visualizing your results from a software platform like MaxQuant. You can see on the left-hand side that there's an organizational principle for whether or not you're looking at chromatography, ion sampling, peptide identifications, contamination, and even some metrics that are more directed towards single-cell proteomics. And there are also some additional items that we'll return to when we do a live demonstration to see how this works. So you may have noticed from this software package that we're visualizing things as distributions. Why are we doing that as opposed to looking at summary statistics like the mean or the median, standard deviation? Those are all very valuable, but I can give you 4 very good reasons as to why you might want to look at distributions. So all of these distributions have the same summary statistics, and they're part of sort of a famous grouping called Anscombe's quartet. They all have the same mean, same median in both of their parameters, and the same correlation between their parameters. But if you look at the distributions themselves, you can see that you might draw vastly different conclusions about the relationship between these parameters. If we just take 2, shown in the top left-hand corner, increasing x corresponds with increasing y. Pretty straightforward. But this is only true up to a point in the top right-hand corner. As you increase x past a certain tolerance, it corresponds with decreasing y. Now this is fine in the abstract, but I think it becomes more important to us as we want to visualize things like the number of identifications over the course of a gradient, because this corresponds to the physical properties of the ions that we're analyzing. You might, for instance, be losing hydrophilic ions due to a stage tipping protocol that you're using, and you want to be able to see that, and you can see that using a distribution. So what's the toolkit that we need to effectively benchmark the performance of our LC-MS system and make sure that it'll work for single-cell samples? The first thing you'll need is a stable sample, which we'll talk about momentarily. You'll need some sort of search software, whether or not that's MaxQuant or PD or your own favorite suite of software tools. And we recommend using DoMS just because we've iterated through this, but you'll need some way to actually visualize and draw conclusions from the data generated via your search software. So if we take a look briefly at the stable sample that we recommend, Harrison touched on this a little bit in his presentation. We recommend a 100x master sample. Now, how do you prepare this? You have 2 different cell types that you have lysed in bulk, and then you serially dilute this into your different sample channels. And in this way, you're able to control for different lysis efficiencies. You've also digested them in bulk so that you're controlling for different digestion efficiencies as well. And then you'll label these. And this is sort of a rubric for, I guess, the peptide input that you would expect in each different channel. So when you inject 1% of this sample, you're injecting a set that corresponds to roughly what you would want to see in your experimental samples. You can see here that we're using 2 50-cell carrier channels made up of different cell types. And additionally, we have about 6 single-cell samples as well, complete with some control wells that Harrison talked about the motivation for in his talk. Now, a lot of times people will just use BSA or some synthetic peptides or perhaps even a HeLa digest to benchmark the performance of their system. But if you really want to see whether or not your system is performing at the level that you would need it to for single-cell proteomics, it's good to use a sample that is as close to your experimental samples as possible. Now, in order to use DoMS to its full capability, you'll need to enable a couple of options within MaxQuant. So within the Global Parameters tab and the Tables subtab, you'll need to enable the output of a couple of different data files, which are MS scans, MS scans, and all peptides. And you also need the Calculate Peak Properties option, which gives you some additional information about where you're sampling your precursor species. And we'll look at that a little bit later. So like I mentioned earlier, there's an enormous amount of information that these software tools can provide you. For instance, the all peptides output file that we see here lets you know every single ion that was seen by the mass spec, whether or not it was MS-MS'd, whether or not it was identified, what the score of that identification was. All this information is there for you. Sometimes you just have to enable it in order to get access to it. And the last thing that you could definitely use to benchmark your performance is this software package that we've developed. It's freely available from doms.slavovlab.net. It's on GitHub. And this includes links to our recent JPR article, also the bioRxiv article. And Albert really spent a lot of time when he was building this website on not only providing you with steps for how to get started, but also for how to actually personalize this. For instance, if there are metrics that you would find useful that are not in the DoMS platform, it's very straightforwardly personalizable. So we hope you'll give this platform a shot and give us feedback on how it works. We love to hear about the bugs because they help us optimize the platform. So what we're going to do now that we've kind of built up a motivation for why to use reproducible samples and DoMS, we're going to talk a little bit about some of the ways that we've used this system to optimize our own system. So early on in the scope series of experiments, we were kind of exploring the whole parameter space, 60-minute gradients, 90-minute gradients, 120-minute gradients, different gradient pitches, different buffers. And once we settled on using a 60-minute gradient and a 250 millisecond MS2 injection time, we kind of realized that there was still some optimization that could be done in terms of where we were targeting our ions. And we'll see this momentarily. So earlier I talked about trying to maximize the probability with which you're sampling that apex, because it's very important for single-cell proteomics. Here you can see a representation of how we were doing with targeting the apex of the elution profile. These are just vertically oriented histograms. On the x-axis, you can see the number of ions that are in each bin. On the y-axis, positive numbers correspond to sampling before the apex. Negative numbers correspond to sampling after the apex. And you can see that for our default instrument settings, 250 milliseconds of MS2 injection time, we were consistently sampling early. So we reasoned that maybe there's a way that keeping most parameters the same, other than MS2 injection time, we could potentially begin to sample closer at or near the apex of the elution profile. And so we reasoned that by expanding this collection window, the MS2 fill time, we could bias ourselves towards this. In essence, just waiting a little bit longer to sample the ions of interest. And you can see that this worked. As we varied from 250 milliseconds to 500 milliseconds to 1,000 milliseconds, we were able to more effectively target the ions at or near their apex. And this had some, I won't say totally unexpected, but this, we were able to draw some interesting conclusions by looking at things across all levels of analysis. So here we're looking at peptide identifications, but we're doing so in perhaps a slightly unusual way. So what we have here is a way of looking at identifications across all confidence levels. So you can kind of get the full distribution. Now, we're looking at things in terms of confidence in terms of PEP. So for PEP as a metric of confidence, lower numbers are better. And then we just have a running tally of the number of identifications that are present at or below that level of confidence. So you can see that for our highest MS2 injection time, for our 1,000 millisecond injection time, we got around 750 confidently identified peptides. For our 250 millisecond time injection time, we got around 300 to 350 peptides. And we won't, I think it's important to say that the apex targeting, we can't say definitively that that's what resulted in the increase in the number of identifications, but the correspondence between increasing the injection time allowed us to both more effectively target the apex and resulted in more confident identifications. So we can also use this platform for diagnosing sample issues, which is, as Harrison mentioned, Murphy's Law holds in every laboratory. And sooner or later, you're going to have a sampling issue or a sample issue, whether or not it's labeling, digestion, et cetera. And so we have a toolkit here to help you diagnose these issues. So we've looked a little bit at the relative reporter intensity as a way to diagnose sample prep issues before. Here we've got a sample with twin 50-cell carrier channels. Previously, we were looking at one with a 100-cell single carrier channel. And how this plot works is that we have normalized all different channels by that channel with the highest median reporter ion intensity, which is usually, hopefully, one of your carrier channels. So just from a quick glance at this plot, we're able to see that our twin carrier channels have comparable levels of peptide input, that we have two control wells, and that this isotopic contamination, or the the impurities within our TMT labels, are visible here. 2 channels down from your 50 Jurkat cell channel, you see that we have some signal, less so than in a true single cell channel, but perhaps it corresponds to about 1% of our 50 Jurkat cell channel, which would be about half a cell. You might also be able to chalk the presence of this signal up to column carryover, because about 1% of your sample may stick around through subsequent runs. And here you're also able to see that in terms of the ratios between your single cell channels and your carrier channels, we're on order where we need to be in this particular experiment. If you're looking for a 50-to-1 ratio on this log 10 relative reporter ion intensity scale, that would correspond to a value of about 1.7. Now we're slightly above that, but we're in the ballpark of where we need to be. And you can see that we effectively quenched our TMT labels because if we had not quenched effectively, and some of the TMT labels had perhaps interacted with our carrier channels upon mixing, you would see very inconsistent levels of reporter ion intensity in the single cell channels. So this is another way for you to assess how well your sample prep went. Now, some additional features that I'll get into through this live demonstration are the ability to export publication quality figures, subset your data, and also track longitudinal performance, but we'll come back to that momentarily. So what we have here is we have DoMS open in a web browser window. I'm going to load a data set for us to take a look at. And all I'm doing is sort of locating on my computer where the max-quant output files are. I'm going to hit confirm. It's going to remind me that I've actually already loaded this, so it's going to skip that. And then I can load the data. There's a little progress bar in the right-hand corner because some of these data files are quite large, all peptides, for instance. So it's good to know where you are in the loading procedure. And then we can come down to the bottom here and perhaps make some more informative labels for our samples. Now, once we've updated our labels, perhaps the first place most people run to after they've done an experiment is to see how many peptides did I quantify. So we'll go to the Peptide Identifications tab. And what we see is between these two replicate injections of 100x master sample, we have wildly divergent results. In one case, we're identifying about 750 peptides at a PEP of 0.01. In another case, we're identifying perhaps maybe around 300. Now, these are replicate injections, so what went wrong? And that's one of the strengths of this platform, is that it allows you to specifically diagnose issues. So in order to begin this process of diagnosing what may have happened, we can take a look at the MS1 precursor intensities for our ions, because perhaps we just injected less of the second replicate. But this doesn't seem to be the case. If we look at the top right-hand corner, we can see the MS1 intensity for all identified ions. And these two distributions are comparable. They may be off by a quarter of a log in terms of their mean. If we scroll down here to just look at the MS1 intensity for all Z greater than 1 ions, those that are peptide-like, because most peptide-like ions are 2+ and 3+ ions. Most of our contaminant species, our buffers, our detergents, are +1 ions. We actually see that of those ions that we sampled from sample 2, we're more abundant than in sample 1. And then another very interesting component of the DoMS analysis platform is that we're able to control our comparisons. In this plot, you can actually compare the peptides in common between your two samples. You can have a controlled comparison. And what we see here is that it doesn't seem to be the case that we injected maybe half a microliter of sample 2 and 1 microliter of sample 1, because we're only off here probably about a quarter of a log. But there's one ion population that we haven't looked at yet, and that's the population of contaminant ions. So if we go to the contamination tab, we can see that there was indeed an issue with sample 2. In these bar plots, you can see the number of ions by their charge state, going sequentially from 1, 2, 3, and greater than 3. Sample 2 contains over 20,000 of these contaminant ions, whereas sample 1 contains under 10,000. And if you look at the total ion current, the intensity of these ions, sample 2 has strikingly higher abundance of these contaminant ions. And one other very useful metric is you can see where these contaminants elute. Now, in most of our sample preparations, you will see contaminants elute at the beginning and end of our gradient. That's typical. But what's atypical is that they would elute over the course of our active gradient, competing with our peptides for charge and also analysis time. So through this series of plots, we're able to kind of identify what the issue may have been in our sample preparation as specifically as possible. We hope to provide this. I mean, this is available, freely available on GitHub, and we hope that it enables your own research, because like I mentioned earlier, optimization really depends on your own experiments and really depends on your own system. So this is customizable in the sense that you can add new plots if there's a metric that you don't see here. I mentioned earlier that you can output these plots as graphics, as PNG or PDF files. And you can also, if you're interested, generate reports that contain all of these plots and share them within your lab and also archive them. So I have an example of one such report here. It contains all the same tabs as the dynamic plot window. It's shareable. It's self-contained. You don't need to share all the different assets, the different plot pictures. And it's quite compact. Additionally, one other thing I'd like to show is that you can use this platform for longitudinal tracking of your own instrument performance. So we run 1x injections, sometimes a daily, a weekly basis, to keep track of how our instrument is performing, to make sure that before we run an experimental set, that it's performing well with our 1x injection of standard. And this is just an excerpt showing you the performance from late March, from the 21st to the 28th. And you can kind of track a slight decrease in the MS1 intensity for the ions. This allows us to see when we need to clean the heated capillary or the front end, for instance. Or you can also look at the chromatographic measurements to see perhaps if there's some sort of clog. That's also a useful metric. So at this point, I just want to take a moment to thank all the people that were involved in this project. So my co-authors, Harrison and Albert, and Professor Slavov, we worked really collaboratively on both the manuscript and the platform itself. to develop something that was more broadly useful than the internal version that we were using in the lab. I also want to thank Emmott for all of the helpful feedback on both the manuscript and the platform itself. I don't know if Tony Koller is here, but he was also one of our best beta testers. He found so many things wrong that we fixed, so many issues. And we also want to be sure to thank Jürgen Cox, because it was during last year's conference, during a breakout discussion section on data quality, assessing data quality and data sharing, that we realized that the broader community might be interested in a tool like DoMS, and that kind of drove us to take our internal version and make it more easily usable by others outside of our lab. And I'd also like to thank the NIH for funding this research as well. So with that, I'd be happy to take any questions. Or you can also feel free to contact me later by email if a question comes up during installation, something like that. So thank you for your time.
Questions for Gray?
Based on your own experience, how would you say this optimizing system is going to work for different samples? How long does it take, and how much effort do you think?
So that's kind of it's a great question, but it's one that is kind of complex, because often you're optimizing multiple things at one time. You might be optimizing the performance of your LC-MS system, but also exploring different carrier channel inputs.
Okay, let me separate. Would you think the mass or HPLC or mass-spec part, if we have optimized for one sample, would that work for other cell type or tissue? Or do you think the sample prep have more variability for different cell type or tissue?
It can be a question of peptide input in some cases. So if you optimize for a single cell set like presented here in the 1X master, there is some, I think, general carryover between that and different single cell preparations, although cell types do vary in the amount of protein that they contain. So you might need a more abundant carrier channel or a less abundant carrier channel. I think it's, we've processed a variety of different samples from collaborators using this 1X to benchmark our performance. And sometimes the variability can come in unexpected peptides that were in your sample, such as highly abundant things like hemoglobin. So I think a lot of the optimization that can be done is in the sample prep process prior to submitting them for analysis.
Hi, great talk, thank you. Did you compare with the same LC system different mass-spec acquisition platform? So using the same LC system, but changing the mass-spec?
We have not. We typically rely on a QE Classic in our lab. We have not hooked our same LC system up to another mass-spec.
So I think Gray said that very well, but let me just make one more point to reiterate that with our preprint, we have shared all of our raw and process data, as well as the parameters that have worked for us. And you're very welcome to use those same parameters and see how they perform for you. But we don't necessarily expect that these are the Goldilocks set of parameters that are going to work for every system. We think that every cell type, every experiment, every LC, every instrument is going to have a slightly different set point. And the intention here is to give you a set of tools, principles, if you will, how to derive the optimal set of parameters, rather than pick a single set that happened to work very well for our samples, and then expect that this will work for every other sample thereafter. So with that, let's thank Gray again. And